So the first part of this, we were looking at the some different ideas about chemical reactions and kind of how they relate to metabolism. Now we're getting into the nitty-gritty of how cellular respiration actually happens, and this is really the story of cellular respiration. So what we're going to do here is actually take you through the actual steps. I'm going to go for purple because why not? So cellular respiration, this is going to happen in three distinct stages. First stage is glycolysis. Glyco as in glucose and lysis as in to cut. The next stage is pyruvate oxidation plus citric acid cycle. One thing I don't want you guys to get hung up on, you could divide these and subdivide them into step upon step upon step. So sometimes people would put this in a diagram and just say it happens in 2 steps, glycolysis and then Krebs cycle, and they kind of lump all this stuff together. Just for these notes, we're going to break it down this way because we're trying to get you the whole story. Then the third step here is oxidative phosphorylation, which is 2 technical steps if we want to get it super technical about it. It's the electron transport chain and chemiosmosis. So those ones, you might even just see this called the ETC or called chemiosmosis. Sometimes people will use these interchangeably for number 3. It's not technically correct, but don't let it throw you off. So this one includes electron transport chain and chemiosmosis. These are 2 subparts of that process right there. Okay, this is an overview of the stages. I'm gonna get you guys a copy of this that is blank. Worst case scenario, if you just look this up, I'm sure you can find ones that are already blank. But this is a big overview of what's going on. Just get you oriented. I'm not going to spend too much time on this. So outside here, this is the cytosol. So that means it's inside the cell. Here's the rest of our cell here. So we got one giant mitochondrion. So there's our mitochondrion, which is just singular for one mitochondria. I love this overview because it shows you all 3 steps. So we have glycolysis, pyruvate oxidation, citric acid cycle, aka Krebs, and oxidative phosphorylation. So this one actually breaks this into 4 distinct steps here. The other thing this does a really nice job is showing you where electrons are going. So remember earlier we said that this is partly about the flow of electrons, which is also known as the flow of energy. And then there's also a flow of matter. So you guys are responsible for knowing both. Yay, lucky us. So electrons, when we say they're carried, it means that there's an electron within here in these bonds. You just can't see it because it's inside the bonds. And then we've got some other stuff going on here too, but I'm kind of all over the place, my bad. But basically we've got our inputs in here. We also have our outputs, so it's telling you ATP, and that's saying, okay, this is what's happening at these levels. So this is how ATP is actually getting created by substrate level phosphorylation, by oxidative phosphorylation. All right, now let's break this down and go into some of the actual nitty-gritty. First of all, the mitochondrion. So when we look at this thing, the inner part here, which is all cytoplasm, that's called the matrix. Then we have the outer membrane and the inner membrane. There's also these folds in it, and these are called cristae. So you could think of these as some girl named Cristae. Hey Cristae, how you doing? There's Cristae. So she's got her smile on. She is in one of the folds here. And then there's this area in here outside of the matrix. This is called the inner membrane space. And so this diagram right here, I would say return to this, guys, because it shows you citric acid cycle going on inside the matrix here. So that's where glucose gets finally broken down. And then also the electron transport chain, which it's pointing at this actual space right here in the membrane, you know, this inner membrane. So a couple of really important things here. I'm going to switch over to a different color that shows up better. Okay, let's go through stage 1, which is glycolysis. Glycolysis, step A here. During glycolysis, sugar, meaning glucose, is split to produce a small amount of energy. This is believed to be ancient. Why? Well, because prokaryotes do it. And they had to do this when there was no oxygen available. Back in the day, when the earth was first forming and living things were first coming online, oxygen was poison. So they lived in a world without oxygen. They still needed energy. Glycolysis, this happens in the cytosol, aka the cytoplasm. And what happens here is that glucose gets partially oxidized. And if you remember back to when we were just talking about oxidation and reduction, so you think about this, glucose is actually getting oxygens added to it. That's one way to think about this, maybe the simplest way to think about it. So we started with glucose here, which is a 6-carbon sugar, and that gets broken in half into 2 3-carbon sugars called pyruvates. The net gain from this, we gain 2 ATP and we gain 2 NADH. This also makes 2 water. So that's another thing to consider as you're going through this. No oxygen is required for this. So this happens without oxygen. Glycolysis summary here, glucose gets broken down, gets turned into 2 pyruvates, which are shown as 3-C, or this chemical formula right here, which if you remember, glucose is C6H, oh my gosh, it is hard to type with this thing, 12O6. So there it is, split in half. Okay, and now this diagram right here is showing you that there was a phosphate group that got added ADP and ends up making ATP. So there were 2 ADPs, there were 2 phosphates, those got turned into 2 ATPs. Also along with this, we had 2 NAD pluses. What would this be? Would this be oxidation or reduction? So you got to think to yourself, is it gaining an electron? In other words, becoming less positive? Or is it losing an electron, becoming more positive? So it's actually gaining an electron, which means that this has been reduced. When in doubt, guys, I don't know what to tell you. Just guess reduced, because it seems like more than half the time they're talking about reducing things. Okay, an output from this is also water, H2O. Okay, glycolysis itself, now we're getting even deeper into that. It happens in 2 steps. This is really worth investigating because you've got to understand that anytime that we have a chemical reaction, you have to put energy into it. I can't tell you how important it is that you understand that. You have to put energy into it. So the first step to any chemical reaction here is energy getting invested into the chemicals. So in this energy investment phase, the cell uses the ATP that it's created to phosphorylate compounds of glucose. And then stage 2, this is the energy payoff phase. So here's what this might look like. Here's a molecule. So we're going to string this along. I'm getting lazy, so I'm only going to draw 4 of these. So these are atoms. Each one of these is an atom. And now we have ATP coming along. And ATP is going to actually donate a phosphate right here. Well, another ATP comes along, donates a phosphate right there. I should have drawn these right next to each other, but at any rate, once it does that, these are really unstable. So it's actually more stable for this compound if this thing just breaks apart. So now this is going to split into a 2-atom molecule, and this one also splits into a 2-atom molecule. So that's what's going on in glycolysis. Only to be more accurate, we've got 3 instead of 2. So that'd be 3 and 3. So there's the energy investment phase. We had to spend ATP in order to break these things apart. Stage 2 is the energy payoff phase. So in stage 2, these 3-carbon molecules, which I'm doing just a terrible job of drawing, they're going to get oxidized. So for each glucose molecule, we end up with a net of 2 ATP. Net means what you have left over. You could also think of net as your profit. So you had to invest some ATP, and then we made some ATP as well. I'm doing a pretty shoddy job of keeping track of my investments here, but basically we invested 2 ATP, and so we gained 4 ATP, which is where we get our net of 2 ATP from. So we invested that, that's what we spent, that's what we got, which gives us this leftover. We also got out of this 2 molecules of NADH. Okay, so this substrate level phosphorylation, this is transferring a phosphate from one molecule to another. And in general, this creates a small amount of ATP. So when we say something has been phosphorylated, that means an enzyme has actually transferred a phosphate to other compounds. So here we have some phosphate going on. So here's the substrate that had a phosphate group on it. So remember in that last slide, I'm going to go back a second here, these guys had the phosphates on them. Well, here we go. There they are, phosphates. And now that phosphate is getting added to ADP. Which is actually going to create our product, boop, and ATP. So now we've gone from a decharged battery, we've taken the phosphate off there, clicked it back onto here, and now we have a charged battery again. These steps, you don't need to know. So you can stare at this for a second and then celebrate the fact that you don't need to know them. However, what you should understand is that each step on here, like they could on a test ask you to take a look at a diagram like this and then discern something about it, like how is ATP used or where's the energy investment phase. So what you're really looking for here is just trying to figure out what the diagram is showing. So in this case, we're seeing different chemicals, like this one's called glucose, then glucose 6-phosphate. So you're seeing a different chemical in each one of these boxes. You are seeing an enzyme name for the enzyme that does this underneath each arrow. And then what this is showing, ATP going into that reaction and coming out ADP, so it's dropping off its phosphate. So right here, this would be the energy investment phase. And then if we kind of skip down to here, you see that we actually have the energy payoff phase. That would be something they could ask. They could also ask you about this right here. They could say, like, during which of these steps is NAD being reduced? And you would have to say, oh, okay, step 6 right here, because NAD plus becomes NADH. So NAD plus has been reduced. So those are two types of questions that they might ask. Okay, we'll stop it right there and start up on our next one.